Wherever you find yourself, and good evening from Australia. My name is Elvis, and today I'm presenting on the topic release of plastics to Australian land from Biosolids end use. And I'm actually based at the University of Queensland. Now, globally, the production rate of plastics has been increasing since 1950, reaching 348 million tons in 2018. But a simple question we should all be asking ourselves is, what happens to all these plastics that we produce? Well, they do get into the environment some way, somehow. Now, over time, all of these plastic products that get into the environment break down to become smaller pieces, to become what we call microplastics, which are plastic particles that are less than 5 millimeters in size. As we study plastics, we are learning that they are everywhere, in the food we eat, the air we breathe, the water we drink, in soils, in wastewater, rain, and even biosolids. Now, wastewater treatment plant has been identified to actually receive plastic particles from the industrial sources, household sources, and then in some countries from stormwater that are actually connected to these wastewater treatment plants. Now, the plastic particles that get to wastewater treatment plants are filtered from the liquid fraction and are retained in the sewage sludge that are produced from the treatment plant. Now, studies have reported that more than 80% of these plastic particles that get into wastewater treatment plant through the influent are actually filtered from the liquid fraction and remain in the sewage sludge that are produced. Now, in countries such as Australia and across Europe, and even in America, these sewage sludge are then treated to become what we call biosolids and then applied to agricultural lands. Now, plastic particles in these biosolids that get to the soil environment can then be actually washed into other environment, and then the chemical additives that are used in these plastics can be leached and then can be taken up by a plant, which can then end up in the food chain, or they can be transported to other environments, such as nearby rivers, which can pollute aquatic environment. Now, in Australia, in 2019, 371,000 metric tons of biosolids were produced. Out of these, 70% were actually applied to agriculture land, which compares to 60% in USA and Canada and 50% in Europe and China. Now, biosolids have been identified to be beneficial, and they've been reported to be able to improve soil conditions, which then improve agricultural productivity. However, they've been reported to be sinks and pathways for plastics that get into wastewater treatment plants, and they've been reported to be pathways of plastics into the soil environment. Now, one study actually reported that up to 430,000 metric tons of plastics were actually released to agricultural lands or soils from biosolid application in Europe and North America. Now, the plastic particles, like I mentioned before, that get to the soil environment have been reported to be toxic to organisms. They've been reported to be impacting organisms' feeding rates, causing mortality, and impacting their reproduction. Now, like I mentioned earlier, in Australia, more than 70% of the biosolids that are actually produced are applied to agricultural land. Now, currently, there are several methods or techniques that are used to actually identify or quantify plastics in biosolids. This include using organic degradation digestion protocols to actually degrade organic matter in these biosolids, and then actually filtering these plastic particles in the biosolid using a vacuum filtration onto a filter paper. Now, studies have gone ahead and used optical microscopy to actually identify these plastics or subject some of these plastics into identification. And then some studies have also gone ahead and picked some of these plastics that are identified on the filters and then chemically identifying them using either FTIR or Raman spectroscopy. Now, these techniques have been used across the world and they've been used in plastic analysis for so many years. However, these techniques provide information on the particle count, the number, sizes, color, shape, but not the total mass of plastics in these samples. And like I mentioned earlier, these techniques are also operator dependent. Something that I will say it's plastic, someone will come and tell me it's not plastics. And they are also size dependent, which can lead to underestimation or overestimation. Now, FTR have actually been identified or reported to be able to identify plastics that are above 20 micron, whilst Raman spectroscopy have been identified or reported to be able to identify plastics that are above 1 micron. Now, it should be known that these techniques are very time consuming. In order to actually quantify the total mass of plastics, irrespective of the particle sizes in a sample, there was a technique that was developed in 2016 by a research group in Australia. And what they did was that they used pressurized liquid extraction techniques to dissolve all these plastic particles in a single sample. And then they actually analyzed the flake that formed from these plastics using an FTIR. Luckily for us in our lab, we have Pyrolysis GCMS. So what we sort of doing was that we thought of combining the pressurized liquid extraction technique with a Pyrolysis GCMS, which then allows us to actually give a mass-based quantification. This technique is actually able to extract all the particle sizes in a sample, which means that you get a total mass of the plastic in a sample, which is better than getting particle counts. And then if you have a technique that's not able to identify particles that are 1 micron, then you lose some of these particles. Now, during the method optimization, we use dark chloromethane at 180 degrees and then 1500 psi, to extract these plastics from the samples using the PLE. And then our method validation was able to extract 7 plastics, which included polystyrene, polyethylene, PET, PP, PVC, PMM, and polycarbonate. Now, the extract from the pressurized liquid extraction was then injected into a pyrolysis GCMS, and then the mass-related concentrations were actually quantified using an external calibration. Now, after validating and then publishing the method, we applied this method to 82 biosolids samples from 82 wastewater treatment plants that were collected in 2016. Now, this treatment plant actually covered 34% of the Australian population in 2016. And then the 7 plastics that we developed the method for were actually quantified in these samples. We also went ahead and took some data from the wastewater treatment plant operators to actually help us in our analysis. 7, now plastics were actually detected in 99% of these biosolids. In one treatment plant, we couldn't detect any plastics. And this treatment plant actually provided us with ash samples from the incineration of biosolid, which suggests that incineration can also actually be used to actually get rid or destroy plastics. Now, 6 out of the 7 plastics were actually detected in these samples. The only plastic that we couldn't detect was polycarbonate. And then the mass concentration range was from 0.4 and 23.5 milligram per gram per dry weight. Now, polyethylene was the predominant plastic by mass, followed by PVC and PET. Now, the per capita plastic release across these 81 sites were between 8 and 877 grams per person per year. Now, for us to understand the sources of these plastics into biosolid, we sort of try to compare the amount of plastic consumed in Australia to the amount of plastic we identified in each of the states in Australia. As can be seen in Figure A, it actually shows that in Australia, the highest amount of plastic consumed, or the major plastic consumed, was polyethylene, which followed by PVC, PET, PP, polystyrene, and PMMA. As can be seen in the data that we got in the left side of Figure A, we found out that the concentrations of plastics were actually increasing at the same rates with the plastic consumption in Australia. If you look at the Figure A, you could see that polyethylene was the highest consumed in Australia, and it was the highest detected in the sample across states, followed by PVC, PET, PP, polystyrene, and PMMA. Now, to actually find out where or what factors were actually influencing the concentration, we tested the significant difference between or the relationship between the per capita mass loads and the size of the treatment plant, and then the per capita mass loads and the states and between the states. But we found no significant difference between these factors. And then there was no relationship between percentage industrial area and the per capita mass loads. What this suggests or tells us is that there could be other factors that were actually impacting or releasing these plastics into the wastewater treatment plant, which then means that we should try and go further and then analyze the sources of these plastics by actually doing a point source analysis. Like, we kind of analyze plastics or wastewater from industries, households, to try and trace where these plastics are coming from. Now, after getting the per capita release, we estimated the amount of plastics that were actually released into the Australian environment through Biosolids application. From the estimation, we found out that 4,700 metric tons of plastics, of these six plastics that we quantified, were actually released into the Australian environment. Out of this, 3,700 was actually applied to agricultural land, and then 810 metric tons were actually sent for other processes, which was included mine rehabilitation, landfill rehabilitation, or external processing. Now, from the 4,700 metric tons of plastics that were released to Australian agriculture soil, each person in Australia actually released 200 grams per year. And then this percentage of plastics that were actually dumped on the Australian environment represented 0.13% of the total plastic consumed in the country. Now, after analyzing plastics in the Australian biosolid, we applied the method to analyze plastics from 1950 to 2016. The idea was to find out if the concentrations of plastic in biosolid had been increasing at the same rate to plastic production. And this is for the 6 plastics that we analyzed. As can be seen in this figure, we found out that the concentrations of plastics in biosolid had been increasing at the same rate with global plastic production, which implies that there's a direct linkage between plastic production and consumption and their release into wastewater treatment plant. Now, specifically for the individual plastic that we tested, we found out that prior to the 1990s, the leakage of plastics into the wastewater or the biosolid was limited, apart from only polystyrene. So polystyrene was the only plastic that we could identify in the biosolid samples prior to 1990s. Then from 1990s, there was an increase in PE, polyethylene, and then PVC, which actually increased from 1990 to 2016. Now, in summary, the pyrolysis GCMS and the PLE method we developed actually provides mass concentration of the selected plastics that we tested, and then the quantitative per capita mass load and emission estimates of plastic types. We were also able to provide data on the quantitative per capita mass load and emission estimates of plastic through biosolid end use. And our method extends our ability to detect plastics in the nano-size range and all plastic particles. What I mean by this is that the PLE extraction method is able to dissolve all the plastic particles that you have in a sample, compared to the spectroscopy and the visual identification techniques where there's a size limitation and you are not able to actually, give a total mass concentration of the samples in our samples. I want to thank the wastewater treatment plants in Australia that gave us the samples. And then I thank my fellow students and staff members at Quiz who helped in the data analysis and sample preparation. I also thank Quiz and UQ for giving my PhD scholarship and top-up scholarships. Thank you.